Okay. Good morning. And unfortunately, yesterday we missed a class, and I am suggesting you to have two double class Thursday for BTL 5 to 7. Please announce it. And today we shall continue our discussion on plant microbiome and their application as uh, biocontrol agent. And we shall share some of our experiences. Yeah, in our last lecture, we discussed how plant and microbiome interact uh, in the environment. And this is really a very nice illustration by Nature Reviews. Beautiful illustration. Here you can see a rhizosphere is a biocomplex where multiple and diverse myriad of microorganisms are inhabiting. They are inhabiting uh, in a high density and high diversity because of getting the plant uh, made carbon source as their energy. You know, plant has the ability to uh, trap uh, solar energy to make chemical energy through the photosynthesis process. I discussed this process in our previous lecture, and you obviously know that process. From the photon, plant use the photon energy uh, in the photosystem 2 to split a water molecule into uh, hydroxyl and an oxygen, which contain extra energy. And that energy through the electron release as an electron, and that electron through the electron transport chain produce uh, ATP and NADPH. And these are the energy in the cell. And those energy in the photosystem 1 used to fix the carbon dioxide from the air to make the carbon containing compound glyceraldehyde 3-phosphate. So if you are asked what is the net outcome of photosynthesis, net outcome of photosynthesis is glyceraldehyde 3-phosphate. Extra glyceraldehyde 3-phosphate pass to the cell for different metabolic pathway to make sugar and many other organic biomolecules, including DNA, RNA. So carbon source in all the organic compounds present in even human, animal, or any other eukaryotic organisms, they are originated from the carbon dioxide fixation by the plant, that is photosynthesis. So photosynthesis is a single biochemical process, very simple process, uh, which is fundamental for the existence of life in this earth. So we are all getting DNA, RNA, protein, amino acid, lipid, carbohydrate, all the compounds. The main source of carbon is photosynthesis. Obviously, a plant needs, some other essential nutrient element like nitrogen, phosphorus, potassium, zinc, calcium, iron, 16 to 17 compounds they uptake from the through the root, and they have the vascular system. Plant have the vascular system, so they transport, and those molecules react with the carbon containing compound and make DNA, RNA, protein, or like that. So they are essential. Even a single essential compound, if missing in the uptake, plant uptake, then plant cannot complete their life cycle. Similarly, essential nutrient element in other organisms also is essential for completion of the life cycle. Otherwise, certain symptoms will be expressed and it would create serious problem. So the carbon containing compound, a large proportion of the carbon containing compound are released, about 40% are released through the root uh, to the microorganisms they are inhabiting in the rhizosphere. So rhizosphere is the rich niche of microorganisms. Uh, in the bulk soil, any field where there is no plant and where there is a plant, if you compare the microorganism, rhizosphere is the richest source, because rhizosphere is the rich source of supplying the carbon containing compound by the plant to the microorganisms, and microorganisms use those carbon energy for their own growth, development, and reproduction. In return, they decompose organic and inorganic compound in the soil, uh, and which is called mineralization or decomposition. For example, organic matter decomposition or like that. And they release the ions present there. That means sulfur as sulfate form, iron is for example, ferrous or ferric form, phosphate is phosphorus is phosphate form, magnesium, magnesium ionic form, potassium ionic form, and plant can uptake those ions. So it is a fantastic mutualistic relationship. So discovery of the beneficial microorganisms, how they crosstalk with the plant, and how they sorry benefit to the plant is very important. But there are some very limited number of microorganisms which are enemy of the plant. They can infect and cause the disease. So there is a competition between beneficial microorganism and pathogenic microorganisms. If we can modulate this microbial world by using beneficial microorganisms, then we can ensure the healthy plant. If you, I have a important information for you. Possibly you know that if you analyze any plant dry matter by using any atomic absorption spectrophotometry or other machine, elemental analysis, you can find 90% of the element carbon, hydrogen, and oxygen, 90 or 95%. That means other essential nutrient elements, 16 or 17, they are basically 5% or 6% or 7%, but they are very crucial. So in the dry matter of the plant, the largest proportion, more than 90% are carbon, hydrogen, oxygen. And if I ask you what are the source is photosynthesis. In the photosynthesis, the compound which is the net outcome of photosynthesis is the glyceraldehyde 3-phosphate, which contain, uh, you know, uh, carbon, hydrogen, oxygen. Anyway, this fundamental understanding surely help you to understand how um, the organisms are interdependent to each other, either animal or fish or any other organisms. All are directly or indirectly linked to the energy harvesting process of the plant. Otherwise, no eukaryotic organisms, even some of the microorganisms, cannot survive in this earth. In our case, we have been trying to discover new, plant-associated microorganisms, which we called plant probiotic bacteria. And in our lab, we have more than 650 plant probiotic bacterial collection. When wheat blast was a serious problem, I asked my team members to check whether any of our probiotic bacteria show any antagonistic activity against the Magnaporte oryzae triticum pethotype, which is the causal organism of wheat blast. Here you can see in a plate, it is called dual culture. Bacteria is cultured here in a streak and mycelial colony here. So they are distant. Here you see there is a gap, but bacteria are secreting some inhibitory substances, antibiotic or lytic enzymes. This is why the growth of the mycelia restricted. Here you see in control completely filled in within 7 days. Similarly, these 4 bacteria, they restricted the growth of the mycelia. That means they are antagonistic. They could be used as uh, biocontrol as a biopesticide. And we checked how this growth is inhibiting. Here in the microscope, you can see many of the cells are lysed and some hyper branching also in the hyphae is seen. And in our past experience, it is clear that some of the antibiotics or metabolites produced by the bacteria, they affect the cytoskeleton of the hyphal cell. And cytoskeleton, you see, we have a skeleton. Every cell has cytoskeleton. Cell cytoskeleton mainly composed of actin and tubulin, and they hold the cell in a certain shape, and they remain just beneath the plasma membrane. So if cytoskeleton is affected, the cytoskeletons are protein affected, then cell cannot grow in its normal shape. Here you see linear growth of tubular growth of the mycelia in normal cases. So this is a very fantastic subject for further fundamental research, how the mechanism of action, as well as to test them in the field, whether they can really control the wheat blast or not. And in our case, we, cultured the bacteria, and after autoclaving, no living bacteria is there, but autoclaved bacterial culture filtrate, when we tested in the media, and we then inoculated the fungus, we found that culture filtrate completely inhibited the growth of the fungus. That means the culture filtrate contains compound or chemicals or antibiotics which are heat stable. This is really very interesting finding. Heat stable metabolite, inhibiting the growth of the fungus. Heat stable compound has benefit for commercialization because their shelf life, they can preserve in normal temperature, even extreme temperature, and that metabolite are stable and they can inhibit the growth. This is very interesting finding. And when we sprayed the bacteria on the wheat seedlings. Here you can see wheat seedlings growing on a plastic pot. Here you see no bacteria were applied. And we sprayed fungal conidia. And what we have seen, 100% of the plants are killed by the disease. And here, no bacteria, no inoculation. That means the normal. But others, 3 bacteria, R3, SL3, and BN8, we inoculated. And we found that bacteria can protect the plant from the, infection by the Magnaporte oryzae. Fantastic protection. And it was at the seedling stage, but wheat blast is a disease of, flowering stage. That means head stage, grain forming stage. So we also need to test it in the field, and we did it later. But what we tried to do, we analyzed the we did whole genome sequences of BTS3, BTS4, and BTLK6. And we compared, searched their genome, and we found them they are producing, they are containing lots of genes involved in biosynthesis of diverse secondary metabolites. Bacillin and macrolactin, these are powerful metabolites produced by the bacillus, and they are bacillus. By sequencing, we confirm that. Here you see lots of, and they also produce different enzymes. They contain the genes responsible for different enzymes, and they are also cell wall degrading enzyme, uh, even gene cluster related to volatile compounds like acetylene and hydrogen cyanide. Hydrogen cyanide, very toxic compound, but there are some differences between the among the three bacteria. Acetylene, this is very interesting compound, small molecule, and we found that all of the bacteria contain acetylene, and it can induce the systemic resistance in the host plant. What does it mean? It means that these bacteria, by using a non-ribosomal polyketide synthase or polyketide synthase enzymatic pathway, they can produce lots of secondary metabolites, and they also produce antioxidant enzymes, cell wall degrading enzymes, and some volatile compounds. And these results are telling us that these organisms have the powerful chemical weapons to ward off or to inhibit the growth of the magna porte orizi. That means the molecular mechanism, we got some very good information. How the phenotypic result we found in seedling assay as well as in the plate assay showed inhibitory activity. And here you can see different kind of secondary metabolites produced by the bacteria. Here, these 4 bacteria we analyzed, and this dark spot means the bacteria produce certain antibiotic, and this is the biosynthetic pathway. So our conclusion is mechanism of biocontrol of wheat blast is through the chemical arsenal. Diverse group of antibiotics are produced by wheat blast biocontrol bacilli isolated from wheat and rice seeds. Very interesting. We isolated them from the wheat seed and rice seed because they are co-evolved in a way, and they are naturally present in the wheat seed, internal tissue of the wheat seed. This is why if we practically apply in the field, that should not be harmful for the nature as well as for the human health. So chemical mechanism, chemical arsenal, antibiotics, and lytic enzymes already revealed by the genomic analysis. Non-chemical means induce systemic resistance in the host plant as volatile compound acetylene is produced. So these are the result, and I will show you later on. In the field test, we also found that complete, not complete, 90% protection of the wheat plant by the application of these bacteria. And bioactive compounds against grapevine, downy mildew pathogen, Plasmopara viticola. This is another dangerous pathogen, and Plasmopara viticola is a biotrophic pathogen, only can grow in the grapevine, angur grass, and cause downy mildew disease. And this disease severely affect the quality of wine produced from the grapes. And this is really economically most important disease, and it has only single host. And when I was in Germany, I developed a method to, because it is an obligate biotroph. You cannot cultivate outside the host. In all the textbook, it is written there. But what I did, I cultivated the fungus it is not fungus, it is an omicity on the lower surface of the leaf on water agar, a 95% humidity and 25 degrees Celsius in the incubator. And then you see, I made a small vacuum cleaner like manual, how to say, tool to harvest all the conidia. Here you see conidia is like this, like grape fruit, and on the conidia fruit like a bunch of fruit, and harvested them. After harvesting, I had a target to get the juice pool, because in natural system, these airborne conidia conidia sporangium, these are called sporangium. Sporangium can move from one plant to another and infect the plant when plant surface is moist. So they release juice pool. I found that they produce the juice pool, and in the medium, and it was a long story because nobody was successful, but I was successful to develop the juice pool in high quantity, and then cystospore, and I also got the germinated cyst. And here, after inoculation, on the leaf, after a few days, how mycelia is coming out to make this type of sporangiophore. So I studied some of the compound, whether they can suppress the zoosporogenesis process, that is the release of juice spore, as well as whether compounds can inhibit the mycelial growth or flagellar motility and cause lysis of juice spore. Because if you can stop at this stage, stop the zoosporogenesis process, that is production of asexual spore, then you can control the disease. Or if you can stop the mycelial growth or motility of the juice spore, because juice spore needs to find the stomata. So if you can stop it, then it cannot infect. And lysis of juice spore means killing the juice spore. That means no chance to get the infection. And by collaborating with a German professor, Hartmut Lach, he is very famous in the world because he discovered more than 100 new antibiotics. I collaborated with him, and I found that staurosporin, and indolocarbazole antibiotic, it can stop the motility of juice spore. And then we design lots of, staurosporin-like compound, different derivatives to get the more active compound. But interestingly, we found that staurosporin, this staurosporinone skeleton is necessary for the activity, but it is weaker active. But if there is a sugar moiety in the structure, then it is higher active. And we also checked the activity of staurosporin, the mode of action in other cells. We found that it is a protein kinase inhibitor. But staurosporin is a protein kinase inhibitor, means protein kinase is a large family of protein kinase A, B, C, or like that. So we checked lots of protein kinase and other kinase inhibitors, and we found that chelerethrine, which is a protein kinase C inhibitor, it is selective for inhibiting the motility of juice spore. And we made a conclusion that the motility of juice spores are inhibited by inhibitors of protein kinase C, suggesting that PKC is involved in the regulating of flagellar motility and peronosporomycetes. Flagellar motility, flagellar motility in juice spore and flagellar motility in human or animal sperm are similar. Interestingly, when we checked the literature, we found that both juice spore and animal sperm motility are maintained by protein kinase C, because protein kinase C is involved in generation of ATP, and ATP is necessary, essential for the motility of the either human sperm, which is very advanced and complex organism, animal, and juice spore is a trivial microorganisms, but they are conserved. But we don't know actually among the protein kinase C, there are a huge structural variation. We do not know exactly what are the differences between the protein kinase C involved in motility of zoospore and the protein kinase C involved in motility of animal sperm. But very interestingly, when we applied the protein kinase C, we also found that motility of zoospore, not only motility of zoospore, but also the zoosporogenesis process is inhibited. And we also found that calcium is involved because calmodulin bind antagonist also stop the inhibited the zoosporogenesis process. So from lots of pharmacological experiment, we confirm that protein kinase C is crucial for zoosporogenesis as well as the motility of zoospore. That means if you can design an agrochemical that can inhibit selectively the protein kinase C in the zoospore, you can nicely control this pathogen. Till now, there is no good pathogen for controlling the. I'm really sorry. Someone is calling. So can you hear me, Shafi?
Yes, sir.
Okay. So here is the data you can see. Staurosporin we discovered from streptomyces, and chelirethrin we got the from commercial source. So chelirethrin can you see can also selectively inhibit the motility of zoospore as well as release of the zoospore. So these two compounds, the result of our discovery, we published in Molecular Plant Microbe Interactions, which is a very famous American journal. So mechanism is motility of zoospores are inhibited by inhibitors of protein kinase C, suggesting that PKC is involved in regulation of flagellar motility in peronosporomycetes. And before that, people didn't know how flagellar motility in the omycetes like Phytophthora, Pythium, or Plasmopara aphanomyces, which are almost in fact every species of the plant in this earth. They are also, this group of microorganisms are also dangerous pathogen of fishes as well as animals. So this understanding would help to design new drug candidate against the this notorious pathogen. When we tested when we tested the staurosporin on leaf disc assay in the laboratory, we found that 2 micromolar concentration, 100% can protect the plant. That means in vivo test in the greenhouse or in the incubator, we confirm that this molecule can protect the plant from the infection. So this is the result, and we published this paper in Molecular Plant Microbe Interactions. And if you need the full paper, I can share with you. So this is all about today's talk. We can discuss some more. Any question?
Sir, I have a question about the wheat blast, especially.
Please care about your, sound. Your microphone needs to be close to your mouth or like that.
Hello.
We cannot hear you clearly.
Um,
Shafi,
yes, sir.
yes, tell me your question.
Especially in case of wheat blast, we need two, one is primary host and another one
Can
is,
you enhance the sound?
sir, it is highest.
Okay.
Um, uh, we need primary and secondary host to complete the cycle. And I read something about when they complete the generation, sometimes they are also mutated, and also new race is developed.
Yes, all organisms, when they adapt to the host, there is a war, host defense, and the pathogen invasion. So mutation is not unlikely. In case of wheat blast, mutation can be taken place, as well as wheat blast has some alternate host, but in Bangladesh we are not sure what are the alternate hosts. Infected seeds, when they germinate, they also carry the pathogen, and in the older leaves the pathogen remains, and they release the conidia, and through the wind it can circulate and infect the plant. So, what is your specific question regarding the chemical ecology?
Uh, so so two hosts are here is necessary. So if we can we can control the one, then it's also another way to control the blast.
I cannot hear you clearly. Uh, uh,
I bear his question. Can you hear me, sir?
yes,
He mentioned that for wheat blast, there is two host is required, primary and secondary host. So he want to interment one host so that the life cycle cannot be occurred, and thus he want to control wheat blast.
yes, wheat blast, primary host is the wheat, obviously. And Magnaporte or IG triticum is specific for the wheat blast. But there are some weed, relative weed, they can also act as alternate host. What they do cannot, devastating. Wheat blast is not devastating for those wild, plants, but they can survive there, overwinter there, and wait for the when wheat season will come. And under ambient environmental condition, that is high humidity and uh, temperature 18 to 13 degrees Celsius and high humidity. Uh, those on those alternate host wheat blast, produce lots of conidia, and conidia through the wind. They are very tiny conidia. They can move from one place to another, even 6-8 kilometers, and make a cloud-like situation. Even in the air trap, we found that, or a rain drop trap, we found the trace of the DNA of wheat blast. So they remain like that way. If heavy, dew fall or heavy rain, that can wet the wheat plant for 10 hours at least, then it becomes, epidemic. Otherwise, it can be a not so big a problem, 5% or 6% or 10% plants affected in last 4 years we have seen. But 2016, there were unusual rainfall in the winter in 8 districts, and that fueled the devastating outbreak, because water is required for germination as well as penetration and establishment of the disease, and not only establishment of the disease but also spread of the disease, because it can quickly, within a few days, it can produce billions of conidia in the infected plant, and through the wind it can spread to other plants. So this is the scenario. But in case of when the symptoms is expressed in the spike, then if you apply and kill the pathogen by using fungicide, no meaning, because already vascular bundle in the spike is blocked. Water cannot go from the root to the growing spike or grain, developing grain. So shriveled grain is developed and severely yield is affected. But if we can use the biocontrol bacteria, as bacteria are living guard, so before the infection, we found that it is very nice. So when flowering starts in the wheat field, we spray the bacteria twice or thrice at 15 days interval, and we found that we can keep field free from the wheat blast compared to untreated, plants. Any question? Rulamin?
Yes, sir. In case of animal, there are so many uh, fungal diseases such as ringworm, which is also a human disease. So, in case of chick or day-old chick, broader pneumonia, which is caused by Aspergillus fumigatus, um, it is a very threat for the chick rearing in the earlier stage. So, can we check probiotic in our lab to control such kind of fungus, Aspergillus or ringworm?
Yes, if you can isolate the aspergillus, you can check the probiotic bacteria. But idea is, in case of animal, you need to have the probiotic bacteria. You need to isolate from the animal source. Otherwise, plant associated probiotic bacteria should not colonize the animal guts or animal body. So best idea is to get the probiotic bacteria from the animal source, and then you test the purify against the purified fungus or other organism. And if it works, then through the feed, you can apply to the animal, and
Thank
hopefully
you,
you
sir.
will get the result.
Thank you, sir.
Any question? If no
No, sir.
question, I have question. You have from now 5 minutes time. Uh, I delivered lots of lectures. So within 5 minutes time, you list 20 keywords. Keywords. For example, wheat blast is keyword. You cannot write this one. 20 keywords that you have never heard from any other lectures in the past. You learned for the first time from my lectures. So 20 keywords you list and send to me through my personal Facebook Messenger. So this is your first quiz exam. I can give you 10 minutes time because 5 minutes would be terrible. So but I shall check carefully because the keywords should be unique to this course, and obviously I'm not. You should be faithful. You should not heard before, and for the first time you learn from this course. So time is started now. 10 by 12. 10, I shall check my inbox. So this is your first quiz exam. Thank you so much.
Okay, sir.